The overall goal of this procedure is to generate induced pluripotent stem cells, or iPSCs, from human peripheral T cells in feeder-free conditions.
This method can help answer key questions in the field of regenerative medicine, including how iPS cells can be applied into clinical use. The main advantage of this technique is that iPS cells can be cultured without the inclusion of possible harmful factors, which reduces the risk of pathogenicity.
The implication of this technique extends toward clinically relevant iPS cell-based therapies because of the less invasive cell sampling combined with the reduced risk of exposing the resultant iPS cells to undefined pathogens.
Though this method can provide insight into iPS cell-based clinical therapy, it can also be applied to other areas such as disease modeling conditions.
Begin preparing activated human T cells by diluting 10 milliliters of heparinized whole blood with 10 milliliters of DPBS. Pipette 15 milliliters of Ficol solution into a 50 milliliter conical tube. Slowly pipette the diluted blood, running along the inside wall of the tube, to layer on top of the solution. Be careful not to disturb the interface. Centrifuge the tube at 400 times G for 30 minutes at room temperature.